Hello everybody, so today we're going to be talking about prokaryotes. Prokaryotes are like organisms, they're a kind of organisms. So let's just say a kind of organism. In order to characterize them, they are unicellular. Well, let's just understand what prokaryotes as an organism means. So they are unicellular, but they also don't have a nucleus. No true nucleus. They have a nucleoid, but it is like a fake nucleus. Fake nucleus. So this is one of the very important things that characterizes a prokaryote. Another thing is that they don't have membrane-bound organelles. So no membrane-bound organelles. These two characteristics or features are the defining characteristics of every single prokaryote. That is what defines them. So make sure that you remember these two very important characteristic of any prokaryotes because it's their identifying characteristic. An example of a prokaryote would be bacteria. And there are like 3 types of classification in the way we can classify classification. The way we can classify. So one is a phenotypic classification. Another is a analytic classification, and the third one is a genotypic. So genotypic classification. A phenotypic classification, as the name suggests, depends on the morphology of the organism. So it can be the shape, the size, the structure. And while we're at phenotypic classification, let us also make sure that we are all on the same page with the structure, which is for example, the structure of the cell wall. There are 2 types of cell walls in bacteria. One is a gram-positive and another is a gram-negative cell wall. So this is an example of what we're talking about when we say phenotypic. It's the shape, which shapes can be rod-like, so like bacillus, They can be spherical, so cocci. They can be spiral-like, so which something like, as the name suggests, like a spiral. That is a bad drawing of, but you get the point. So which are called spirula. And then there are other shapes of bacteria which are just shaped as nothing. They're just like a mixture of everything. And that is a category as well. So that's what we're talking about when we say phenotypic classification. When we say analytic classification, we talk about the cell wall type. So let's go here and say the cell wall type, the cell protein type, because every organism, even in prokaryotes, have different proteins, and they have different functions of proteins. So the protein types, the organelles they have are different, so in both prokaryotes and eukaryotes. So this is like a general understanding. So cell wall type, protein types, also like the enzymes that are present in the cell. All of this comes under the analytic classification of bacteria. Just in case people are not familiar with the gram positive and the gram negative cell walls, I have made another video for it, and it's again on the channel. So separate video. So if you need to brush up on that, I would suggest that you either go after this video or stop this video and watch that video, so that we are all on the same page. Okay, next up is the genotypic classification. So genotypic classification, as the name suggests, is the classification of the genetic level, if you want to call it that way. So the DNA or RNA present, RNA, the bacterial chromosomes, and so on and so forth. One thing I would like to point out while we're at the subject of genotypic classification is that bacteria have an organelle called plasmids. Plasmids are DNA strands that form a circle, or a circular DNA strand. Circle. And so instead of the normal double helix, double helix strands, the plasmid DNA is the same double helix, but it's just in a circle form. So these are plasmids, and it's important to know that their replication is independent. Replicate independent. of the normal cell chromosomes. So plasmids are like extra chromosomes. Not chromosomes, my bad. They are extra sets of DNA which are completely independent of the regular chromosomes. Or DNA. So this is one of the unique features of a bacteria, that they have plasmids. To review this whole thing again, prokaryotes are unicellular. The definition of a prokaryote is that it doesn't have a true nucleus and it doesn't have membrane-bound organelles. An example of prokaryotes is bacteria, and bacteria are classified into 3 classifications. One is phenotypic classification, where we consider the morphology or the physical structure of the bacteria, like shape and size and structure. So here we have all the shapes of the bacteria. And then moving on to the second classification, it is the analytic classification, and it has the cell wall type or the cell protein type or the enzymes that are present in the bacteria. And here this relates this relates to the gram-positive and the gram-negative cell wall. And if you need to brush up on that again, watch the separate video that is on the channel, which will help you understand that better. And the last classification is the genotypic classification. As the name suggests, it is about the genetic level of classifying bacteria. And here we look at the DNA and RNA, and we look at the chromosomes, and plasmids are a part of the genotypic classification. What are plasmids? Plasmids are circular DNA strands that are independent of the regular DNA in the cell. So this is all for the prokaryotes video. I hope this solves your doubts, and I will see you in the next video. Thank you for watching.